Hello student, today I am going to discuss somaclonal variation and its application in plant tissue culture. This video is for second semester and sixth semester BTech Biotechnology students. So today's topic is somaclonal variation. Please note that this video will be of 5 minutes and afterward it ends, and then I will prepare next video and then I will add. So it's a series of video. Okay, so now we are going to start our topic somaclonal variation. Now, as the name suggests, somaclonal. Okay, if we look at the front camera, okay, the name suggests somaclonal variation. Okay, now all of us know that variation means differences that we observe in a population. So if we observe a population of plant, or we observe even population of human or population of animals, then we find a numerous differences among themselves. So we call it as a variation. Now, what is the meaning of somaclonal? Okay, now if we look at the word soma and clonal. So now soma means somatic. Okay, and somatic stands for 2N diploid cells which are present inside a plant. Okay, so there is a somatic cells, and clone is something a copy. Clone is something a copy. Now, if we look at the what we say micropropagation or aim of tissue culture is mass multiplication of plants, and this mass multiplication of plants is obtained with the help of micropropagation. So now from a single explant we produces n number of plants. Suppose we have produced 1 lakh plant. Now there is a large population of plants, and if we find some differences among these plants, these 1 lakh plants, though these plants have been produced from a single explant, so they are clones of each other, and clone generally refers to a genetically similar copy. So purpose of tissue culture is produced n number of genetically similar copy, that is clone. But still among these large population there is variation. There is a exception. Theoretically, clone is a genetically similar copy. Theoretically, tissue culture should produce genetically exact replica of the explant or mother plant. But practically, this does not happen. In nature, we find vast differences among these plants. And that's why these plants have been produced from the somatic cells, that is 2N haploid cells. In micropropagation, we either use shoot or either use any part of the plants. Okay, so these variations are observed in the clones, particularly somaclones. So we call them as a somaclones because they are produced from the diploid cells. So these variations we are going to study under the we are going to study these variations with the help of PPT. So now all of you should be very much clear about the meaning of somaclonal variation. Okay, so these are the differences that has been observed among the population of plants produced under in vitro condition. So we call it as a somaclonal variation. It is that simple. And soma stands for somatic cells. Somatic cells are diploid in number, and clone stands for a genetically similar copy. So in this genetically similar copy we find as few differences. We call them as a somaclonal variation. And tissue culture has wide applications, wide influence, okay, on this somaclonal variation. So now in next video we'll look after the slide, okay, of this somaclonal variation. So this is a part 1 video which I am now going to end it. Thank you.